Please feel free to type your introduction in the chat as well, in case you're having audio issues. We'll read it out to the rest of the participants. All right, next, can we hear from Rajesh? Hello, Rajesh.
Yeah, good evening. This is Dr. Rajesh from Shastra University. I'm working as an assistant professor in MSc Data Science Department. And again, couple of students of my students were going to join here. So the main motive of attending this webinar is to get more insights about healthcare data analytics, since we have a course called healthcare data analytics. So that is our motive to attending this webinar along with my students. Yeah.
Thank you so much, Dr. Rajesh, for joining. We hope that today's webinar will give you the required insights to help students advance their.
Thank you.
Yes. And let's hear from the next person. Vasheka, can you please introduce yourself? We are curious to know how many of you are from life science background and how many of you are from computer science background or any other fields. Please do let us know in the chat. Yes, Vasheka.
Yeah, this is Vashika here. Hello, am I audible?
Yes, Vasheka, you're perfectly audible.
Yes. I'm from the science background. And currently, I'm pursuing data science, the PG degree. So, like, we are undergoing in Shastra University. And a few of my... And I guess so that's it. We are like, I did my UG in mathematics and now I'm currently pursuing MSc Data Science. And I'm from Chennai and I study in Shastra, Chennai.
Oh.
And yes, that's it.
Oh, thank you so much, Vasheka.
Thank you.
And we have one introduction from the participant named Viresh in the chat box. He is from a life science background. Can you also let us know where are you from? Are you from Shastra University as well? Or in the meantime, let's move on to the next participant. Can we hear from Aishwarya?
Hello.
Yes, Viresh.
Hello. I'm Viresh. I'm currently doing PhD in Oribra University. So my basic background is life science and I'm working in the environmental DNA. So basically in my molecular biology, and my other projects are in the transcriptomics. So this is just my background.
Thank you so much, Viresh. That's a pretty interesting background. And let's hear from the next person. Who is the next person? Kanya. Can we hear from you as well? Kanya, are you here with us? Okay.
Yes, ma'am.
Yes, Kanya. Can you also introduce yourself? You can let us know about your educational background, research interest.
I'm Kanya from MSc Data Science Department, where I was a mathematics student, BSc Mathematics.
Okay. And what are your research interests?
Can you come again? I couldn't hear you clearly.
Sorry, could you please repeat that?
No, I wasn't able to hear you.
Okay, I was asking, what are your research interests?
For now, I'm just open for anything. I'm not fixed in.
Oh, all right. So I hope that today's webinar will give you some idea into the various ways that the bioinformatic application works and help you get started in the field. So let's go on to the next person. Anthony, can we hear from you? Okay, Anthony, perhaps you can introduce yourself in the chat box if you're having audio issues. Then the next person is Charles Usuji. Can we hear from you? Can you please introduce yourself? We just have 2 more minutes left. We'll just quickly finish up with the introduction part and then start with the webinar.
All right. My name is Charles Usuji from Nigeria. I'm a graduate student of biotechnology. So I'm happy to connect to OmicsLogic for this seminar, for this workshop. Thank you.
Thank you so much, Charles. Then the next person, can we hear from Dr. Rabia? Okay, I guess Dr. Rabia is having audio issues as well. Let's move on to the next person, Dr. Yinka. Can we hear from you? Can you please introduce yourself and let us know about your educational background and research interest? Please feel free to introduce yourself in the chat box as well. We'll monitor the responses in the chat as well. And along the way, if you're having any queries during the webinar, you can post them in the chat box, and our team is here to address any of the queries you might be. Okay, Dr. Rabia and Dr. Yinka seems to be not available. Next person, can we quickly hear from, yes, Dr. Rabia, hello. Can you please introduce yourself? Yes, hello, Santosh. Okay, please do keep your responses coming in the chat box. Our team is here to monitor any of the queries you might be having, as I mentioned. So without further delay, I'll just quickly start with today's webinar. Hello everyone, a very warm greeting to one and all. My name is Srigavi, and I would like to welcome you all for today's webinar on OmicsLogic, Bioinformatics, and Data Science Training Program. In this webinar, we will provide a holistic insight into our innovative training offering designed to empower participants at various skill levels and interests. And these programs span a wide spectrum, catering to diverse learning needs. And these programs span introduction to bioinformatics, genomics, transcriptomics, as well as biomedical data science. So more on this will be covered in our upcoming slides. So before that, let me take a brief moment to introduce the team behind these programs. So we are India and US-based bioinformatics company dedicated to empowering researchers, clinicians, as well as students with the advanced skills necessary to navigate and excel in the complex field of omics data analysis. And our commitment towards bioinformatics and data science training extends globally through collaborations with esteemed institutions like Louisiana Biomedical Research Network, National Association of Idea Principal Investigators, Georgetown University, Tauber Bioinformatics Research Center, IIT Jodhpur, NIPA Hyderabad, and more. From specialized training courses to research services that provide expert guidance and support, we are committed to providing comprehensive solutions for all your omics data needs. And additionally, our internship and fellowship programs offer unique opportunities for students to gain hands-on experience and mentorship, while our degree programs with leading institutions provide a pathway to advanced education and specialization in omics data analysis. And through partnership with coordinators worldwide, we have successfully conducted bioinformatics training and research programs for over 26,000 individuals from different countries, having benefited from specialized programs in oncology, infectious diseases, precision medicine, neuroscience, and biomedical data. At the core of our training program are the OmicsLogic learning management system, which is a user-friendly learning platform for students, researchers, and faculty to learn, analyze, and interpret high-throughput data. Here we follow a student-centered learning model where the online programs are divided into different levels based on the background of the participant, that is, beginner, intermediate, and advanced. And during the course of the program, students will be required to go through the mentor-guided lecture recording, online quizzes, as well as several online courses and example projects that include topics ranging from introductory bioinformatics, genomics, transcriptomics, metagenomics, epigenomics, data science that includes bioinformatics using R and Python. And throughout the duration of the training, our team closely monitors and tracks the learning progress of the student through checking the number of learn points achieved, courses completed, program learning progress, and so on. And for learning and practical analysis, we give two options for the students. For instance, you can learn bioinformatics without any coding. And for those interested in opting the non-code pathway, they will be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. The platform includes demo pipeline as well as data management and analysis cloud infrastructure. So more on this will be covered in the upcoming part of today's webinar as well. And for those interested in learning to analyze data through coding, we provide the Google Colab notebook, which help you develop coding skills while learning popular packages in R programming and Python to visualize, annotate, and analyze complex patterns in the omics data. And now here's a quick glimpse into the various ways that some of our students have leveraged these training resources to advance their academic and professional careers. As an example, Okafor, with a background in computer science, acquired fundamental knowledge in bioinformatics through his enrollment in the Introduction to Bioinformatics program that helped him to leverage his technical expertise and work on projects on genomics and transcriptomics during his internship with an industry. And another student outcome is that of Ali. He has a microbiology background, and having secured genomic data analysis skills at the end of the training program, it helped him to successfully apply for various PhD positions under supervisors engaged in infectious disease research projects. Catherine, on the other hand, coming from a molecular medicine background, was able to transition to data science after gaining necessary skills in transcriptomics and R programming. Rima, a systems medicine graduate student, was able to gain necessary skills in Python programming that helped her to work on a machine learning project on diabetes. So all of these student outcomes as well as projects are available on the learning management system that I was talking about, which is the OmicsLogic learning system. And so to help you navigate that, I'll just quickly walk you through on getting started. Just give me a minute. So meanwhile, if the participants have any queries, please feel free to type it in the chat. Meanwhile, I'll share the link to the OmicsLogic learning management system that I was just showing you on my side. We do have a couple of responses in the chat box and introductions from the participants. We'll quickly go over them. Yes, Sakshi is from a MTech in computational biology background at IITD. And what about the rest of the participants? You can also introduce yourself. And meanwhile, let me quickly take you to the class. So this is the OmicsLogic Learn portal, which I was just showing you in my slide. And in this learning management system, the first step is to create an account. So to do that, you can simply visit the link I'm sharing in the chat box, which is learn.omicslogic.com/signup. Okay. And for those participants who already have created an account on the portal, you can simply sign in using your existing login credentials. And for those participants who are joining us for the first time and have to explore the portal, I'll quickly walk you through the steps. So once you visit the link, click on the create an account option that you see here. And here you will be asked to enter a few details, such as your name, email ID, password, your university, and so on. Or you can also sign up using any of your existing social media handles. So this could be your Google account, Facebook account, Apple account, GitHub account, or even Twitter. All right. And for now, for those participants who already have created an account on the portal, simply click on the login now option that you see here, enter your existing email ID and password, and simply click on. So now with that, I'll just pause for a couple of seconds for the participants to sign up, and I'll monitor the responses in the chat box. If you have already created an account on the portal, please put a done. And once you have created an account on the portal, please put a yes, so that I get an idea about those who are already familiar with the portal. All right, Uzair has already signed up. Dr. Rajesh has also signed up. Sachi has also signed up. All right, perfect. Many of them have already signed up. Saloni, Faizan has signed up. Akila has signed up. Aishwarya has also signed up. And if you're facing any technical issues while trying to log in, you can mention that in the chat box as well. And our technical team will be able to address any of the issues that you might be facing. All right, so with that, I'll just quickly proceed to navigating the portal. So if you have any questions, if you want me to pause in between, you can let me know. So I'm going to sign in using my existing login credential. So once you sign in, the very first interface that you see is the courses page, right? And the first step is to set up your profile on the portal. To do that, simply head over to the section on the blue bar that says your name and your, okay? It will direct you to the account settings. Now here, when you click on the profile button, you will be able to update your profile. So what are the various sections that you need to update? Make sure that your name is in proper capitalization and with proper spelling. This one, when you're completing the courses and downloading them, this name will be printed on them, right? And please add a brief bio about yourself. You can mention about your educational background and research interests and link your social media handles. So once all of this is done, it will be showing 100% profile completion. Now, these are the various sections under your profile. Under the activity tab, our team will be able to monitor the learning progress that you have been making and assist you in case you're stuck. Under the courses tab, it will display the various courses that you're currently taking and the learning progress that you have been making. And under the programs tab, it will display the program that you have enrolled in. Under the certificates, once you have completed the required assignments and quizzes, you'll be able to download the required certificate for the courses as well as the programs under this section. So, and on the blue bar on the top, you'll be able to see two more sections that means courses and programs. So under the courses tab here, it displays the various library collection of the courses and example projects that we have on the portal. And here on the search bar, if you enter a keyword that you're interested to learn more about, let's say genomics, it will display the relevant courses and example topics. Alternatively, you can also simply click on the required button, Explore, under the different sections, genomics, transcriptomics, machine learning, and it will display it. And under the programs tab, you can explore the various innovative training programs that we are offering at the moment. And here also you can simply enter the keywords that you're looking to learn more about, or simply using the tags that have been given under the learn more about section. So that was all about that I wanted to cover in my part of today's webinar. So now to explore the programs in depth, what is the program structure, what are the various training resources that you get access to, and various student outcomes, I'd like to pass on the stage to Sonali. Over to you.
Thank you so much, Sri Gauri, and a very good morning, afternoon, or good evening in whichever part of the world you are. I welcome you today to the webinar where we'll be discussing about the spring programs. So today I'll be briefly taking you through 4 major programs that we are about to begin with in the month of March. The first one is Introduction to Bioinformatics. Another one is Genomic Data Analysis, RNA sequencing or transcriptomic data analysis, and then we'll also go through the program Biomedical Data Science using Python. Before I begin with what topics we'll be covering in each one of these training programs, I'll just quickly talk about the goal of the program and how these programs are structured. So the goal of these programs is to provide you training in bioinformatics and data science, the tools and techniques that could be used to analyze the data, enabling independent research guided by mentors and peer example projects or research works. That will develop a growing community with shared passion for data-driven research and appreciation of citizen science that will enable you all, be it students, clinicians, or faculty of all the backgrounds, to develop novel and independent research using latest technology in life and data sciences. So during this program, you all will have an access to a wide range of valuable features and resources. Firstly, the program will offer you online mentor-guided training and research experience that will ensure the participants receive personalized guidance and support throughout their learning journey. There are mentors and subject matter expert videos available providing further insights and expertise in the field. And to enhance the practical aspect of the program, live bioinformatics and technical support would also be provided that will enable participants to receive real-time assistance with any challenges they might encounter. You'll also get the access to the T-Bioinfo platform. That's a comprehensive platform that facilitates hands-on practical experience in bioinformatics. And also the program integrates open source alternatives that will incorporate popular scientific tools and algorithms that are widely used in the field. And to reinforce the learning and assess progress, practice quizzes and graded assignments are also included. And finally, upon successful completion of the program, the participants will receive a certificate that would recognize their achievements, enhancing their professional credentials, just as Sri Gauri showed you how you can access the certificates on your profile. So these comprehensive offerings would ensure a rich and rewarding experience for participants that would equip them with the necessary skills and the knowledge in bioinformatics. So these programs that I'll be just talking about, they are arranged in 3 different levels. So if you're a beginner and eager to learn the fundamentals, or an experienced researcher who is looking to enhance your skills, you'll have these different levels of expertise that you can pick up based on your requirement. So the first one is the beginner level, for the ones who just want to begin understanding about a specific type of field. Then you have the intermediate level, and then you have the advanced level as well. The 3 levels are different based on the number of sessions, based on the duration, and obviously the topics that would be covered in each one of these levels. There are more insights that are given on the program page. I'll take you through that towards the end of my presentation. Okay, so let's pick one of these programs first of all, that is Introduction to Bioinformatics. So when the session started, we asked you what your backgrounds are. And we got to hear from some of the students that they had a mathematical background as well. currently pursuing data science though. And as there is a boom in the big data, and specifically in biomedical science, I definitely understand the eagerness and urge of students to learn about big data analysis, to learn about biomedical data analysis. Before you get started with biomedical data analysis, you really need to understand what are the different types of omics analysis? What are the possibilities out there? What different types of data is present? And then you start working on the tools and techniques that you can use to extract insights from this data. It's very important for you to understand the data as well before you really begin analyzing it. So this program is perfect for those who come from a non-bioinformatics background and are eager to learn bioinformatics. This program will set a stage for you, a foundational stage for you, and help you understand the different types of omics fields. All right. So for the first question that I want to ask is, what is bioinformatics and how do we really use it? So bioinformatics is that science that stores both lots of complex biological data that you can analyze it to find new insights that could be used in many different ways. And today's biomedical research isn't simply about publishing one set of results and hoping that the other researchers would read it. It's about finding the patterns in all forms of data that can link everything that is out there to provide new insights that we can only spot if we can see the bigger picture. Bioinformatics lets us bring together the data from lots of experiments in one place so we can ask those big questions and find the answers. And such data from different sources is available. Rather, the availability of such rich sources of data changes the nature of biological research itself, as it is rapidly acquiring the character of a data science. And so we have amongst us data scientists here as well, or future data scientists indeed. So who uses bioinformatics and who is it for? The answer might have been statisticians, computer scientists, or engineers. But the truth is that more and more biologists and clinicians are now expected to understand and also handle their own data to publish and succeed in basic and translational research. In fact, biology itself is rapidly acquiring the character of a data science. Now, as biology is rapidly acquiring the character of a data science, billions of data points on genes, proteins, and other molecules are compiled in large files and they're systematically studied. So, analyzing this data leads to more knowledge and understanding about living organisms that includes human health, biotechnology, crops, and livestock. So, in this training program, we'll give you an overview of bioinformatics for students, biologists, and clinicians. We'll understand how the influx of data, associated methodologies, and data-driven research makes bioinformatics for life sciences as prominent as the emergence of computers for the tech industry. So this field is based on high-throughput technologies that generates billions of data points on genes, proteins, and other molecules which characterize biological systems and phenomena. Now, these data points, they are compiled into repositories and they can be systematically studied to understand, interrogate, and even establish trends for many applications. Now, where is this data available? There are many repositories out there that we'll explore throughout the training program. One such repository is NCBI that most of you might have heard about, National Center for Biotechnology Information. It has a collection of peer-reviewed articles, biological data sets, and projects on various topics. So as you can see, in the past several decades, this resource has been growing exponentially. There are over 80,000 publicly available data sets in just one part of NCBI that is called GEO, and the submissions are increasing every day. GEO stands for Gene Expression Omnibus, by the way, and such data repositories contain data sets on thousands of genes in each sample, with tens or even hundreds of samples per data set. So you can imagine the amount of data you have there lying in the repositories all ready for analysis. So the program is designed to highlight the role of big omics data that is shaping modern biomedical innovation, where molecular data is used for precision diagnostics, big data integration, and machine learning are transforming personalization of treatment selection, and innovative algorithms allow for unprecedented capabilities with drug discovery, design of large molecules, and even drug repurposing. In this program, we'll cover some of the important technologies and its applications in today's world. The participants will be able to access bioinformatics resources along with expert and mentor support to get the help with learning process that will help you learn the transformation of biology into data-driven biology. As a result, you will learn about the basics of all different omics types, starting from the field bioinformatics, what it's all about, what type of data you deal with in the field of bioinformatics, be it genomics, transcriptomics, metagenomics, single-cell transcriptomics. You will be introduced to these different fields. We'll not dive much deep into each one of these fields, but we'll introduce you to these fields, what type of data you get, where the data is stored, what type of analysis can be performed, and a taste of the analysis would be given in this specific program. And you'll also learn about the tools and the techniques that could be used to analyze these different types of data types that you would be introduced to. So that was about Introduction to Bioinformatics Training Program. The next program that I want to discuss about is Genomic Data Analysis. So you all know what is DNA, how is it organized, how it is used. Let's discuss how to analyze the DNA code. So we will look in this specific program how to analyze the DNA code, how to look for the variants, how to identify the variants, how to perform comparative or functional analysis, in short, if I speak about this program. So how did we come up with this specific program, or where did the field of genomics come up from? So you all know that the year 2020, it marks a 70-year mark since the discovery of the structure and the function of the DNA molecule. It took almost 50 years. It took almost 50 years from that time to sequence the first human genome. And soon after the technology developed, it evolved rapidly. And by 2015, the 1000 Genome Project brought a significant number of whole genome sequences to the research community. And in 2018, over 1 lakh whole genomes were sequenced. And today there is an ever-increasing number of genomic, transcriptomic, proteomic, and metagenomic datasets that are available that describe subcellular processes with highly detailed digital data. The suffix "om" that is being used to define various fields of omics in molecular biology and bioinformatics refers to totality. For example, the word genome was first coined in Germany in the beginning of the 20th century. The word genomics refers to the totality of the genomes or studies of the genomes. So genomics is an interdisciplinary field of biology that focuses on the structure, function, evolution, mapping, and editing of the genomes. And a genome is an organism's complete set of DNA that includes all its genes. And in contrast to genetics, which refers to the study of individual genes and their roles in inheritance, genomics aims at the collective characterization and quantification of all of an organism's genes, their interrelations, and influence on the organism. So that is in total of what we'll be going through in the program. Now, to understand how genomics is used, we need to understand the analysis approaches to DNA code, specifically how we can identify changes that characterize them and interpret their significance. You all know about different types of variations and mutations that are there. I'll not get too much in depth about it right now. But to summarize the genomic variation, they can be of several different types. When analyzing the genomic data that we are looking to find and characterize the changes in the DNA code and associate them with phenotype, here are some acronyms that you should remember about genomic variation. One is SNV, SNP, CNV, and then you have the genotype. SNV is a single nucleotide variant, nucleotide change that may or may not be present in this population. Basically, this means any change from the reference genome found in a given DNA sequence above the level of technical error. SNP, that is single nucleotide polymorphism, when a nucleotide is known to be altered in a proportion of the population, typically considered to be prevalent in approximately 1% of the population. Then you have CNVs, which are copy number variations. That is when a gene is encoded in multiple copies, but the number of copies varies. And such changes, as well as the characteristics of these variants, are commonly referred to as the genotype. Now, why did I dig a little deep into these different acronyms that I said that you must know about? It's important to us because when we look at the genomic data that we get from these different sequencers out there, it's important for us to look at these different types of variants and understand what sense they make at all. But the question is, where is this data available? All of those we have today as participants might not be researchers in labs and might not have the access to data directly. So if you're students and you want to learn how to perform genomic data analysis, can you access the data? Yes, you can. There are multiple repositories out there where the data is available. The question is, what type of data you need to download, how to extract the data, how to prepare the files for analysis, and then perform the analysis and gain insights out of it. So in the program, we'll go through this process. What are the repositories that you can explore? Some of them are definitely GEO, Bioproject, that you can extract your data from. What type of data you need to look at when you have to select a data set that you will be working on, how do you extract it, and then how do you prepare a file for analysis? What analysis you have to do for what type of research questions that you might have in your hand. And then there are several databases as well that you can use to interpret the insights of the results that you get from your analysis. So what are those databases that you can use for interpretation of your results to understand what type of variants you have, or what is already known, and what novel insights do you have from your analysis? That is one of those things that we'll cover in the training program. Some of the databases that I have shown on the screen here are Cosmic, ClinVar, and DBSNP, where we can use these databases to annotate the clinical variants that we get from our analysis. Now, the question is, what type of data formats do we need to deal with when we talk about genomic data analysis? So we will talk about the flow of the program would be, we look at the type of data that we get from the different sequencers. What could be the different file formats? There are FASTA files, there are FASTQ files, and then you align them, you get some SAM and BAM files, and then you use a GTF or the GFF files to look at the gene features of the genome. And then you have the VCF files for variant calling, and then you have your results that you can interpret from, right? So what are these different types of files? What do they look like? How do you interpret the information that this file contains is what we'll go through in the training program. All right, once you have the data, what next? So to analyze such raw data, there are different tools for genomic data analysis that are being used. And these include alignment of reads and full sequences, analysis of relationships between the sequences and their evolutionary divergence, measurements of gene expression, relationship between the sequences and the structures. It can be somatic or germline variant calling through germline or somatic variant calling methods. On the Bioinfo platform, there's an example of a big data platform that can be used to perform all of these analysis and many other using an intuitive user-friendly interface. We'll not just dive deep into the pipelines for an understanding of the workflows, but we'll also go through the open source tools and softwares for different types of analysis. We will also discuss about the methods for differential variant calling that can help us compare multiple samples for associations with the phenotype. We look at the different types of sequence alignments, and then we'll also perform a hands-on with multiple sequences on the server and on other tools and softwares. And when the analysis is performed with FASTQ files, there are several important steps to consider, such as pre-processing, mapping, variant calling, and annotation. We look at the workflow. We understand what type of analysis is done at each step. We will also construct phylogenetic trees by aligning those multiple sequences, tracing how the observed species we capture can represent a common source, origin, and even evolutionary time that separates multiple samples in a study. And such phylogenetic studies have yielded important information on how a pandemic or even a disease of a single individual cages with time and gives rise to novel pathogens. We will use the example of viral outbreaks like SARS-CoV-2 or Ebola epidemic of 2014-15 to understand the analysis. We'll also dive deep into genome-wide association studies, which researchers use to groups of, for example, if researchers are using two groups of participants, people with the disease being studied and similar people without the disease. Now, sometimes this can also include certain variations of disease or conditions that is being studied. Researchers can obtain DNA from each participant, sequencing their whole genome, and if certain genetic variations are found to be significantly more frequent in people with the disease compared to the people without the disease, the variations are said to be associated with the disease. And the associated genetic variations can then serve as powerful pointers to the region of the human genome where the disease-causing problem resides. So we'll dive more deep into the jivas in the training program. We'll also look at some case studies. We'll pick up a case study on breast cancer and try to understand the variants in TP53 and BRCA gene and try to interpret the clinical variants that we obtain. We'll also look at the downstream analysis of these variations that we find through the analysis. We'll go through various examples as well in the training program. And as I mentioned, one of those examples will be the TP53 gene. We'll try to understand how the different variants leads to the different functional characterization that comes up because of these different variations. Okay, so that is what we will be covering through in the genomics training program. Now the question is, some of you are definitely up for the coding path. Some of you just want to go through the analysis, perform the analysis, learn about the workflow, and have your analysis done quickly and are not real code lovers, I would say. So the ones who would like to obtain the coding path, we'll take you through the R code path where you will learn how to perform genomic data analysis using R. If you have multiple sequences, how do you align them? How do you construct the phylogenetic tree? If you have multiple sequences from different patients and you want to align them to the reference genome and look out for the variant and then clinically annotate the variant, try to understand if it is pathogenic, benign, it can cause a disease, or if it's not harmful, we'll take up the R code path for you to help you learn phylogenetic analysis and also variant calling through R. So that was about genomic data analysis. Next is transcriptomics for biomedical data analysis. Now, in this program, we'll discuss about RNA-seq data analysis, and we'll briefly introduce you to the field of single-cell data analysis as well. So why RNA-seq analysis? So one of the major challenges in RNA-seq analysis is related to accurate, reproducible, and effective processing of large-scale data sets that represents gigabytes or even terabytes of data. And such large quantities prevent many to gain practical hands-on feel for RNA-seq. So the training program will start with an introduction to NGS, first of all, and then we'll cover the principles behind it and its applications. We'll also take you through the different resources or repositories from where you can extract transcriptomic data, and then we'll follow the path of gene expression quantification, where you generate a gene expression table and then you perform further exploratory and downstream analysis. So there are various repositories that we will be exploring in the training program. One of them is GEO, TCGA, Firebrowse, SRA, Expression Atlas, ENA, GPI is there, and also Human Protein Atlas. We'll take you through these different repositories and databases as well, where we'll teach you how to extract the data, how to prepare your files, and then how to perform the subsequent analysis. You'll also be introduced to different exploratory analysis techniques and downstream data analysis through differential gene expression and biological interpretation. and function analysis through pathway annotation. And participants will also get a range of analytical tools and understanding of these different tools to apply to their own research. And by the end of the beginner level, you will have a good understanding of key concepts and the techniques that you can use in transcriptomic data analysis. Now, as in the genomic data analysis program, I told you about two paths, no code path, code path. So for code lovers, we definitely have R in Python. We'll pick up R and learn how transcriptomic data analysis could be done using R, because we have wonderful packages in R. So we'll be focusing on R for transcriptomic data analysis, from differential gene expression analysis, or I should say from exploratory data analysis to differential gene expression analysis to functional characterization. And to follow the no-code path, you have the pipelines, and also we'll take you through a simple Excel sheet, how you can perform differential gene expression analysis, prepare your data, and then perform the downstream analysis on Excel sheets, how to perform different statistical tests on Excel sheets, and then extract insights from there. And for the no-code path lovers, we will take you through the statistical analysis applications, and then how you can dive deep with the results that you get using different tools and databases from where you can get insights, be it Enricher, David, Reactome, or GeneMania, or X2K Web, from where you can get the insights into the pathways, networks, and gene ontologies. Next is the intermediate level, where we'll cover about the advanced techniques such as pathway annotation, network visualization, and regression analysis as well. And you will also learn about machine learning techniques for unsupervised and supervised analysis for RNA-seq data that will include clustering and classification methods, how you can use these different tools and techniques that you have for classifying or for working with the RNA-seq data. So you will be taken through the process of or the workflow of RNA-seq data analysis. We'll go through the path, no-code path, and the code path as well. It's not that we'll not take you through R if you are following the no-code path. You will be introduced to each and every path, and you can proceed with the one that you are comfortable with. And we also go through different tools and techniques, data science tools and techniques, and machine learning tools that you can use to work with your RNA-seq data. Last but not the least is the Biomedical Data Science in Python program. Why this program? Because the demand for skilled data analysts and scientists who can derive meaningful insights from biomedical data has never been greater. And that's where this program on biomedical data science in using Python comes into play. So in today's rapidly evolving world of biomedical research, the generation of big data has become the norm. And every day, vast amounts of valuable data is being produced that holds the potential to revolutionize our understanding of diseases and also pave the way for groundbreaking medical discoveries. But the sheer volume and the complexity of this data presents a significant challenge, and that's where data analysis comes in. So this program is designed to equip you with the knowledge and the skills that you need to excel in the field of biomedical data science. And through a combination of practical assignments in the training program and the resources, you will embark on a transformative learning journey that will empower you to unlock the potential of high throughput biomedical data. Now, in this program, because we are starting with basics of Python, and then we talk about the different types of analysis that you can do. We'll talk about the basics of Python, then we talk about exploratory data analysis, drawing different plots, and then statistical analysis and the downstream analysis. But because we start with the Python directly, in the beginning, we'll have two lectures, first of all, where we'll just introduce you to different fields of omics, be it genomics, transcriptomics, or metagenomics. We introduce you to these different fields, epigenomics as well, so that you understand what type of data you will get, and then you learn Python in depth, where you can handle these different types of data. So in the program, you will start by understanding the basics that I just mentioned, such as data types, variables, control structures, functions, and modules. Some of you might have a good understanding about it. It will be a good revision for you. And then you'll dive into data processing and cleaning using powerful libraries like NumPy and Pandas. Armed with these skills, you will move on to exploratory data analysis, leveraging visualization libraries like Matplotlib and Seaborn to uncover the patterns and the relationships that are hidden within your data. Additionally, you'll also gain an understanding of dimensionality reduction techniques and machine learning using scikit-learn. And as you progress to the intermediate level, you will deepen your understanding of clustering and advanced machine learning techniques, specifically tailored for biomedical data analysis. You'll also explore clustering techniques like k-means, edge clustering, and spectral clustering using Python clustering packages like scikit and scipy. Many times, students have these questions, when to use which tool. So it's very important for you to understand what these tools are, what do they do, and then you obviously work on the code block to gain hands-on experience with with the capabilities of these tools. So we'll dive deep into the basics first of all, and then we'll build upon it and have a hands-on with the code block. You'll also learn about model validation techniques and evaluation metrics for supervised machine learning models. You'll also delve into advanced machine learning techniques and learn how to apply them to real-world biomedical data sets. So if you are one of those biologists who is feeling stuck in getting started with coding languages, we have it simplified for you. We'll take you through the paths. I'll just take you through those parts of the coursework on the portal shortly. So from visualization of the data to exploratory analysis and hypothesis testing, the practical coding assignments would be given to you, which will help you understand how the code elements that will be a part of any data science project you end up working on in your future have to be dealt with. So that was all that I wanted to cover about these 4 different training programs. Now we discussed about these training programs, but one question that we often get from our students is that they're interested to start, they're interested to learn all these different types of omics and analysis as well, but they're not sure which program to pick. So if you are a newbie in the field of bioinformatics, we'll definitely motivate you to start with the program Introduction to Big Data Bioinformatics so that you're introduced to these different omics fields. Based on your interest, then you can pick a specialized program. If you already have your interest specified, then you can pick the program directly. Be it genomic data analysis, where you will learn about variant calling, phylogenetic analysis, GWAS, the major topics I'm just talking about, and you'll learn about the tools and techniques that can be used to work with genomic data. We'll also dive deep into ML and AI for genomic data analysis, understand what is population genetics and what are the applications of genomics, and most importantly, ethical, legal, and social implications of genomic studies. And if you want to dive deep into RNA-seq analysis, you want to understand how to work with gene expression tables, and then you can enroll for the transcriptomic data analysis training program, where you will learn from the very basics. You start with the FASTA files, you learn how to generate a gene expression table. Some of you are also were also interested to learn Bash, so we'll be introducing that shortly as well, where you will use Bash to generate the gene expression tables. And then once you have the gene expression tables, what next? Exploratory data analysis. Before that, data preparation, then exploratory analysis, dimensionality reduction techniques you learn, you'll get a hands-on with the different techniques, differential expression analysis, pathway analysis, network analysis, gene ontologies, and then the PPIs maybe, right? You'll also learn about machine learning techniques and also go through the code path R for transcriptomic data analysis. So if this program is of your interest, feel free to enroll in this program. Last but not the least is Python. If you want to get a good hands-on with the computing language Python, then this program is the best for you that I just went through, where you will learn about from the very basics of Python. We'll give you an understanding of omics data as well in the first 2 lectures, then basics of Python, and then you dive deep into how to explore data, statistical analysis, and then the ML process, right? So that is how you can maybe choose a program that you would like to get started with. Still, if you have any questions, feel free to schedule an exploratory call with us, and based on your interest, we can guide you. Now, the next question is, what after training? You completed your training. What next? Next, you can work on a research project, apply your learning on a research project. This research project can be submitted as a dissertation to your college or as a report to your university. Or if you just want a publication, maybe for pursuing higher studies, you can work on a research project and then write a research publication for higher studies. It could be a PhD application, or it could be a master's program that you wish to apply for. And so that could be the path. You complete the training program, then you work on a research project, and then you write your report or publication. And for your research, you will be guided by the mentor in one-on-one sessions for your research project, where you will be assisted on your research problem. Whatever type of research work you are working on, you will be assigned a mentor accordingly, where you will discuss with the mentor the objectives of your research. The mentor will resolve your queries, look at your progress, and guide you on the next steps. Okay, so the training flow is like this, where you get started with an introduction to bioinformatics, then you learn about these different omics analysis, and then you learn about these different tools and softwares that could be used for omics analysis. Those who are interested to work on a research project, there are various specialization tracks that you can pick to work on. I just wanted to talk about this because we have received lots of questions about what type of research projects you can work on. So there are different omics data types that you can work on with in these different fields, be it oncology, precision medicine, infectious disease, metabolic disorder, or neuroscience, system bio, agriculture, space omics, or cheminformatics. So you can pick any one of these specialization tracks and explore the possibilities of working with different types of data that you will learn about in your training program. Now, I would like to take you through the OmicsLogic Learn portal and tell you about each one of these programs through the training program page. So this is the portal. Sri Gauri has given you an overview already, so I'll not be going in much depth of it right now. But if you click on the programs here, in the program section, you have these training programs that are up to be started in the month of March. Python, into Bioinfo, Transcriptomics, and Genomics. You also have Cheminformatics and Metagenomics. If anyone is interested, you can scroll through the programs and feel free to enroll in those programs too. Okay. Now, in these training programs, we cover the courses that have been mentioned here as supplementary material. So don't get confused between what are courses and what are programs. These training programs include a mix of resources. They include the session tutorials, lectures by mentors, and also the coursework that you have to go through as a part of a specific lecture. Right now, I'm not a part of this program. That's why it's not showing up. Once you become a part of the program, you can see a session lecture. You can see the resources associated with that lecture and some additional material. Sri Gauri, can you please quickly add me to this specific program so that I can show the participants what access they get to the program? Now, thank you, Sri Gauri. Now, there are different levels in each one of these training programs that you can enroll in for. Shubhangna will explain you how these three different training programs, three different levels vary and how you can enroll for it. Now, each level has these different sessions like this, and the resources would be put together. Meanwhile, Sri Gauri is adding me to this training program. I'll quickly take you through the Bioinfo server and give you a glimpse of a demo pipeline to show you how the no-code path works. If you want to learn the workflow or the flow of analysis for a specific type of data that you are working with, then what the pipeline would look like, what results would you get. So this is the Bioinfo platform where you have different areas of analysis. You have transcriptomic analysis, genomic analysis, cancer bio, single cell, virology, metagenomic. Then you have ML and pipelines as well here, supervised, unsupervised. GWAS, phylogenetic analysis. All right. I'll give you just a quick demo on one of our educational pipelines. If you click on this drop down, you get these demo pipelines. These pipelines are actually educational pipelines that we have put together, and great explanation of these pipelines, what type of analysis was done, what results do you get, is also given in the educational project here that you can go through. Sorry, I'm looking at the programs. If you go to the courses, here you have these educational projects. Okay, so these pipelines have been explained in these specific projects. Let me open up one of these demo pipelines and show you how the analysis is done. So you upload the data. Now this is a demo pipeline. That's where the data is already uploaded at the backend, and I just need to start the pipeline and select the tools for analysis that I need to use. So because it's RNA-seq data, I have the FASTA-FASTQ files. I use Genomic to trim off the adapters and PCR clean to remove the duplicates. You need not understand all of this in detail right now. I'm just showing you how easy it is to use a pipeline. You have these tools and you just upload the data. You have to set up some parameters when you run the pipeline that you'll be taught about. And then you click on the tool and click on the next button, and then you end the pipeline. We'll dive deep into what these tools are, what they do. Many of you might be new to all of this. We'll explore and learn more about it in our program. So you get a multi-QC report, how to interpret a multi-QC report. What is this report, first of all? And then you have these different visualization plots. You get your results here that you can download. You get a gene expression table. A gene expression table shows you the expression levels of genes in different samples. And then you have these plots as well that you can download, the PDFs that you can download. I'll not be going in much depth right now, just giving you an overview. Meanwhile, this loads, I'll just explain the code path. For the code path, what we basically do is you have these different courses on the OmicsLogic Learn portal. Okay, I'll just take you through the Python course as we talked about it. So in the Python course, this course is covered as a part of your training program. In the Python program, you will be given lectures. Along with that, you have the lessons of the Python coursework that you have to go through. Now, inside the coursework, you have these different lessons in the flow of the curriculum that we'll be following. And inside these lessons, you have the code block explained. I think I have a poor internet connectivity today. Maybe that's just taking some time to load. Okay, so let me open this. So once a specific lesson opens up, you will be.
So Sonalika, you have a subscription issue. So I think we have to provide the
Yes,
subscription at
so I
expired.
opened the free lesson. I think let me stop sharing and let it load, and then okay. So once my screen is shared, my internet connection goes bad. I don't know why. Okay, no problem. So once the lesson opens up, you have the code block explained, you have the syntax, you have the explanation of the code block, and you also have a practice Colab that you can go through and learn from. Yes, we have it up. Let me reshare my screen now. So this is what you have inside the lesson. You have the explanation of the packages that are being used. You have the syntax given. You have the other code blocks given. Then you have the explanation of the code blocks, what type of plots you will get, and then how do you manipulate the code blocks to get different types of visualizations. That is how you can go through each one of these lessons. And each one of these code blocks and concepts have been explained in the training program as well. Towards the end of the lesson, towards the end of each lesson, you have a practice code block section. Inside the practice code block section, you have the Google Colabs. So we motivate our participants to go through these code blocks, practice these code blocks on Google Colabs, so that they can share it with us whenever they have any errors, and we help them rectify the errors that they might have. So that is how we practice learning different omics analysis using R or Python. Okay, so that is what I wanted to take you through. And meanwhile, our results are also up. So these are the results that you get from the pipeline. All of them are interactive results so that you can go through and extract insights to the results and also update it in your report. So you have a heat map here where you can see the gene expressions here for each and every gene in different samples, abnormal gene expressions, up or down regulated genes here. And then you can also, this is a volcano plot where you can look at the different types of genes that you have that are up or down regulated. Let's not go too much into the concepts, but the motive here is to show you that you get the results from the pipelines. It's very easy. You just need to know the workflow, what type of data you are working with, what parameters you need to set for each and every tool that you are using. And when you get the results that you can interact with and interpret your insights from the results.
So,
So
Sonalika,
that
there is a
was,
question in the chat. Can you look into the chat?
yes, sir. Yes, you will definitely get the access to the data that you will be going through to practice Python. In the coursework itself, Isha, we have given different types of data examples. In our GitHub repositories, we have different data examples, be it genomic data or RNA-seq data, and you'll be working with these example data types, first of all, to understand how the analysis is done. And we'll also train you how you can extract data from repositories. So you can extract your own data and then you can use the code blocks, the Python code blocks. It seems you're interested in Python, the Python code blocks to extract insights from your data. I'll hand over the stage to Shubhanga now to give you to help you understand how you can register for the program. And in the meantime, I'll just quickly load my program and show you how the resources will show up after Shubhanga is done. So Shubhanga, over to you. Thank you.
Thank you, Sonalika, for such a great demonstration. So I'll be sharing my screen quickly and will explain you the details how to get registered to the training programs. So hello everyone, I'm Shubhanga from OmicsLogic. So as you all know, OmicsLogic offers training in 3 different progressive levels, as Sonalika told you already, in beginner level, intermediate level, and advanced level. So beginner level is for the person who are without a background in computational biology or bioinformatics and are interested in these domains. These are the levels which can provide an easy landing and a curated path to become an expert in the field of bioinformatics. So as you can see in the different training programs, I'm just taking an example of Introduction to Big Data Bioinformatics training program. So this is the beginner level, which will have these sessions and each session objectives, as you can see in the portal. These sessions details are included there. So there will be different sessions, 6 sessions we have in beginner level. Then we move on to intermediate level. So intermediate level covers important data analysis, stats application, and NGS, omics, and downstream analysis using machine learning, creating creative and standard visualization process for you. So as these levels progress, sessions increase, the number of sessions increase, and in the intermediate level, we have 12 sessions in this Introduction to Big Data Bioinformatics. Mind that I'm taking just an example. And in different training programs, different sessions are included. Then moving on to the next session, we have is advanced level. That is for the participants or students who wants to work on a specialized research fields. These training programs provide a special skill sets to them to cover the technologies and newest omics research and frame a research question through these skill sets that they have gained during these training programs. So this was a general overview of different levels of training programs. Each training program includes a specific feature and specialization. So now moving on to the registration process, you can directly enroll through our portal. You just have to click on Enroll Now, and the pop-up will appear to you, where you can purchase a subscription based on your interests, whether you want to enroll for 1 month duration. The beginner level is for 1 month, intermediate level is for 2 months, and the advanced level is for 3 months. So based on your interest, you can enroll for that. Or otherwise, you can contact me on communication@omicslogic.com. I'll paste the email ID on chat box as well. And we have special discount options also available for the students who have enrolled in the webinar. You can change the pricing from here to USD if you are an international participant, and see the pricing here on the portal itself. And if you want a specialized discount through this webinar, you can directly reach out to us at communication@omicslogic.com. So now I wanted to update you on our upcoming event that is happening at OmicsLogic. This OmicsLogic workshop is specially designed for the participants who wants to learn NGS data analysis. This is a 2 days live workshop with 12 hours of live session, and is a great opportunity to students who wants to learn next generation omics technologies. So this workshop will be conducted on 24th and 25th February. We'll have several sessions, and its fees is also there. For undergraduate and postgraduate students, fees is only 2,000 INR, including 18% of GST will also be application on the registration fees. And for postdoc and researchers, the fees is 3,000 INR only. And for industry professionals, fees is 5,000 INR only. So you can register for this workshop as well if you want to learn the NGS analysis. As you can see on my screen, the various modules that will be covered during the workshop. I'll share the brochure with you all, and I'll paste the registration link in the chat box as well, so you can know more details about this workshop. So I'll be conducting this registration process. So if you are interested, you can reach out to me on communication as well. So that's all for today. And if you have any questions, you can reach out to me on communication@omicslogic.com. I have pasted the registration link for the workshop in the chat box, and I'll also paste my contact information and contact number in the chat box. Thank you. And if you have any questions and queries, you can reach out to me.
Thank you, Shubhanga. There's a question by Dr. Rabia. Can you please address?
Yeah, sure, ma'am. Yes, ma'am, you can attend. Not physically, these are virtual workshops, so you can register to the workshop and we can contact you and give you the details of this workshop. I have pasted the registration link in the chat box itself. Or you can email us directly to communication@omicslogic.com. And I'm also sharing you the WhatsApp community link. You all can join there for any further updates, because we are regularly posting the updates there also. So if any new workshop is coming, any new event we are conducting, you can join our WhatsApp community as well. Anyone who wants to ask any further questions or wanted to get more details, please feel free to ask your queries.
No, Matthews, we are not working with ecological data here, but you can be more specific and share more insights with us with what type of data you're interested to work on, and maybe then we can assist you further on it.
those participants who are interested in specific training programs, you can simply email us and we'll share with you more details. So please mention the training program of your interest in the subject line, and we can share with you the curriculum, associated training resources, and so. And if you have queries right now, you can simply unmute yourself and ask, or put it in the chat box. No, Ajay, we do not have training programs for proteomic data analysis. We have for genomics, transcriptomics, and metagenomics. Can you let us know what are your interested training programs based on the topics that we have covered? Yes, Mariam is interested in Python, and I'm not able to understand any free project access. We do have a few free courses available on the portal. We'll email you the link. Please share with your email ID with us, and we'll share with you the required. Python Transcriptomics, if you are interested, we can share our screen and talk more about the training program resources. Thank you, Faizan. Thank you, Mariam.
Along with your email IDs, you can mention the training program so that we can assist you for the same. Thank you.
I have pasted the WhatsApp community link in the chat box. Feel free to join our WhatsApp community as well.
We'll wait for a couple of minutes for the participants to share their training program interest. You can share your email ID and the training program of your choice, and for those interested to get on a 15-minute exploratory call with our team, you can email us requesting for a short Zoom meeting as well to help you decide on a training program of your choice. Okay, thank you, Chef. All right, then if the participants have no further queries, we'll end today's meeting. We hope to see you all in the training program. And once again, if you have queries, please feel free to reach out to us at communication@OmicsLogic.com, and we'll reply back. Thank you so much, everyone. Have a great day.